Chapter 8. Biochemistry and Nutrition. Most daily activities in the animal facility involve chemical reactions, from the cleaning solution that's prepared to sanitize an animal room to the drug administered to an animal and the anesthetic used during procedures. In this chapter, you'll expand your knowledge to include the chemistry of compounds that occur in living organisms as part of their metabolic processes. This is called biochemistry. Biochemical compounds. Biochemistry is the study of the chemical compounds and the complex chemical reactions that occur within a living biological system. These compounds include inorganic molecules such as water, elements such as sodium and potassium, and the major organic biomolecules which include carbohydrates, proteins, lipids, and nucleic acids. The study of the chemical regulation of metabolism, the molecular basis of genetics, and the chemistry of immune responses are important in many areas of basic and clinical research in genetics, molecular biology, and medicine. Impairment in the regulation of biochemical compounds can result in disease. Ketones, for example, play an important role in diabetes mellitus. Diabetes is a condition that results from the body's inability to regulate the use of glucose by the cells. Glucose uptake requires insulin. In a diabetic, the body cannot produce enough insulin or it cannot use the insulin that is provided. In place of glucose, the body metabolizes fat stores for energy. This causes an overproduction of ketones in the body, which results in a condition called ketoacidosis. If left untreated, ketoacidosis can lead to diabetic coma or even death. Similarly, the absence of vital biochemicals such as minerals or vitamins can be equally bad for an individual's health. A deficiency of dietary vitamin C, for example, can result in a condition called scurvy, which is characterized by bleeding gums and tooth loss. Vitamin C is necessary for the synthesis of the structural protein collagen, which is an essential component of blood vessels. At its most severe, scurvy can result in neurological disease and death. Carbohydrates are the most important energy source for the body. Cellulose is one carbohydrate that makes up the supportive structures in plants. Carbohydrates contain carbon, hydrogen, and oxygen atoms. The most basic forms of carbohydrates are units of simple sugars known as monosaccharides. Other classifications of carbohydrates include disaccharides, which are two monosaccharides joined together, oligosaccharides, which are 3 to 10 monosaccharides linked, and polysaccharides, which are hundreds of monosaccharides combined to form a large compound. Mono and disaccharides are commonly referred to as sugars. Polysaccharides are also known as complex carbohydrates or starches. Some polysaccharides are bound to proteins or lipids as structural components of cells. Sources of carbohydrates in animal feed include grains, such as wheat, corn, or oats. Monosaccharides are the building blocks of the other carbohydrates. Glucose, fructose, galactose, and ribose are examples of common monosaccharides. Glucose, which is also called dextrose, is the primary source of energy used by the body for all metabolic processes and can be converted to glycogen, which is a polysaccharide, for storage in the body. Other important monosaccharides are ribose, a component of RNA, and deoxyribose, a component of DNA. These carbohydrates contain 2 monosaccharide units per molecule. Maltose, sucrose, and lactose are important disaccharides. Lactose, found in mammalian milk, consists of 1 unit of glucose and 1 unit of galactose. Sucrose, or table sugar, is obtained from sugar cane or sugar beets and consists of 1 glucose unit bonded to 1 unit of fructose. Maltose, the major carbohydrate in malt, consists of 2 glucose units bound together. An oligosaccharide contains only a small number of monosaccharide units, about 3 to 10. Oligosaccharides are components of glycoproteins, which are compounds made up of carbohydrates and proteins, and glycolipids, which are an oligosaccharide attached to a lipid molecule. Important roles for glycoproteins are cell-to-cell communication and self-recognition by the immune system. Blood types are characterized by the carbohydrate portions of glycolipids on the surface of cells and the carbohydrate portion of serum glycoproteins. Polysaccharides are complex carbohydrates because they contain many monosaccharides per molecule, often 300 to 500, thus making them very large. They may be arranged in long chains that include branches that form a complex structure. Examples of polysaccharides include starch and glycogen, which serve as energy storage in plants and animals, respectively. Amylose is a major component of starch in plants and can contain several thousand of the monosaccharide glucose. Both starch and glycogen are digestible. They are hydrolyzed or broken down to simple sugars by digestive enzymes beginning in the saliva and continuing in the stomach and intestines. Some polysaccharides are not digestible. Cellulose, for example, is a polysaccharide used by plants as their structural material. It cannot be broken down by mammalian digestive enzymes. However, some species, like ungulates, rodents, and lagomorphs, have intestinal microbes that can digest cellulose, so the animal can access cellulose's nutritional value. Figure 8.1 in the manual illustrates different types of carbohydrates, which includes fats, oils, and waxes, share the common physical property of being insoluble in water but soluble in some organic solvents such as chloroform. Lipids include a diverse group of organic compounds such as fatty acids, phospholipids, and steroids. Lipids are the major component of the plasma membrane and form a sheath called myelin around many nerves. They also store energy in the adipose tissues of animals. Some hormones are derived from lipids, for example, prostaglandins, sex hormones, and cortisol. Lipids contain long chains of hydrocarbons, which are groups containing carbon and hydrogen attached one to the other. Lipids may be classified by the type and number of carbon chains or categorized using other criteria. For example, if the lipid contains a phosphate group, it's called a phospholipid. Common types of lipids in biological systems include fatty acids, which can be saturated or unsaturated, glycerides, such as monoglycerides, diglycerides, and triglycerides, non-glycerides, such as steroids, for example, cholesterol, and complex lipids, such as lipoproteins, which include a protein portion, and glycolipids, which include a carbohydrate portion. Chemically, fatty acids can be described as organic acids consisting of a chain of hydrocarbons. The type of bond between carbon atoms define if the fatty acid is saturated or unsaturated. If the fatty acid is saturated, carbon is bonded with other carbons in a chain. All carbons in that chain, except at the ends, are bound to 2 hydrogens. The carbon on one end is bound to 3 hydrogens and is called a methyl group. At the other end, the carbon is bound to an oxygen and an alcohol. That carbon is known as a carboxyl group. An example of a fatty acid is shown in Figure 8.2 in the manual. The term saturated is due to all carbons being saturated with hydrogens. That is, they are bound to as many hydrogen atoms as possible. Another important type of bond in fatty acids is a double bond between 2 carbons, as in Figure 8.3 in the manual. With this kind of bonding, the carbons attached to the double bond have 1 less hydrogen, thus a total of 1 hydrogen each. Because this type of fatty acid is not saturated with hydrogen, it is called an unsaturated fatty acid. If the chain contains 1 double bond, it is called monounsaturated. If there are multiple double bonds between carbons, the fatty acid is called polyunsaturated. There can be up to 3 double bonds in an unsaturated fatty acid. Animal fats generally contain less than 60% unsaturated fatty acids, while vegetable oils contain more than 80% unsaturated fatty acids. The most common saturated fatty acids are palmitic and stearic acids. The most common unsaturated fatty acids are oleic and linoleic acids. Some fatty acids cannot be synthesized in the body and are called essential fatty acids. Linoleic and linolenic acids are known as essential fatty acids, and thus they must be included in the diet. Skin and hair coat problems in animals are often associated with deficiencies or abnormal metabolism of cholesterol and lipoproteins. Triglycerides are the main constituents of animal fats, such as those found in butter, red meat, and poultry, as well as vegetable oils such as corn, peanut, safflower, and olive oils. Recall that tri means 3. Triglycerides contain 3 fatty acids, various types, attached to a glycerol molecule as shown in Figure 8.4 in the manual. Triglycerides help solubilize fatty acids so they can be carried in the blood and distributed in tissues. Triglycerides are a type of fat carried in blood. Measurement of triglyceride levels in a serum sample is a common method of assessing lipid metabolism to the group of lipids called steroids and is also measured in serum to help determine normal lipid metabolism. The chemical structure of cholesterol is shown in Figure 8.5 in the manual. Cholesterol and triglycerides are insoluble in blood. They are transported in the blood by attaching themselves to proteins, forming a complex called a lipoprotein. Some lipoproteins transport the triglycerides and cholesterol consumed in food from the intestines to other tissues, while others transport internally synthesized triglycerides and cholesterol between tissues or to the liver for storage. Shortly after a meal containing a significant amount of fat, lipoproteins are visible in a blood sample. The plasma of a centrifuged sample will have a cloudy appearance instead of its normally clear color, a condition called lipemia. Lipemia is normal in this circumstance and quickly resolves as the lipids are transported, for example fats and oils, begins in the stomach. In many species, such as pigs, dogs, and rabbits, the stomach secretes an enzyme called gastric lipase. Rats and mice have glands in the tongue that secrete lipase, which is also active in the stomach. This lipase hydrolyzes 10 to 30% of the dietary triglycerides in these species. Lipase continues activity through the stomach and into the small intestine, where other enzymes assist in breaking down the triglycerides into the component parts of glycerol and free fatty acids. Bile is secreted by the liver into the small intestine to emulsify lipids and some vitamins. This allows them to be absorbed through the wall of the small intestine. This allows lipids to enter into the energy pathways of the body or to be incorporated into body structures requiring lipids. Proteins perform the widest range of functions of all the biological molecules. Because of this, transgenic models are used to research the role of specific proteins to help our understanding of disease processes. Proteins are grouped according to their role. The major categories are as follows. 1. Structural proteins, which make up things like the fibers inside muscle cells and the outer coating of some viruses. They're often laid down in sheet-like layers or spiral strands. Many proteins combine shapes of sheets and spiral strands in different parts of their molecule. Keratin and collagen are examples of structural proteins. Number 2. Enzymes are biological catalysts which perform work of chemical reactions in cells. Enzymes may be solubilized in the cell cytoplasm or contained in cellular organelles, in tissue fluids, or in digestive juices. Enzymes may also be integrated in the membranes of cells and their organelles. Lipase is an enzyme that enhances the digestion of fats. Proteases are enzymes that have a role in the digestion of proteins. The synthesis and degradation of biologic compounds require the help of enzymes. Many enzymes require minerals and inorganic molecules to function efficiently. Number 3. Regulatory proteins, which help turn certain chemical reactions on or off, such as in the regulation of gene expression. Like enzymes, these proteins may be solubilized or integrated in membranes. Typically, regulatory proteins are intracellular. An example is the anti-inflammatory cytokines, which are produced by monocytes and macrophages to repress the body's immune response once infection is resolved. Number 4. Transport proteins, which are membrane proteins that transport substances into and out of the cells. Hemoglobin carries oxygen to the cells. Lipoproteins, such as low-density lipoprotein and high-density lipoprotein, transport cholesterol in the bloodstream. Number 5. Hormones, which regulate body functions. There are 2 classes of protein hormones. The first class is monoamines, derived from aromatic amino acids such as adrenaline, dopamine, or serotonin, and the second class is peptide hormones, made up of amino acid chains such as insulin or thyroid-regulating hormone. Many hormones travel via the bloodstream and enter tissues to exert their effect. Number 6. Protective proteins, which act as bodyguards, protecting cells from attack by foreign substances. Antibodies and the complement cascade are protective proteins. See Chapter 12 for more information. Number 7. Toxins, which include substances of individual proteins, each with a specialized function. Together they assist in and carry out the range of biological functions required for life. Collagen is the most abundant protein in the mammalian organism, comprising about 1/3 of the total protein content. It's an important structural component of bone, tendons, cornea, and other connective tissues. Amino acids are the building blocks of proteins. The sequence of amino acids in a protein is coded by genes. As their name suggests, amino acids are organic acids with an amine group which contains nitrogen. Amino acids bond together to form either peptides or polypeptides. Peptides are short chains of up to about 100 amino acids. Polypeptides, also called proteins, are longer chains of amino acids. There are only 20 common amino acids, but there are a very large number of possible combinations of these amino acids to form proteins. The order of the amino acids in the protein is referred to as the amino acid sequence. For example, enkephalins, which modulate pain sensation, are peptides that are made up of a chain of 5 amino acids. Beta-endorphin is a peptide containing 31 amino acids. Albumin is a large protein found in serum. Amylase is another protein that is an enzyme that aids in the digestion of starches and other polysaccharides. Table 8.1 in the manual lists the common amino acids proteins are represented in print by their amino acid sequences using either their 3-letter or 1-letter abbreviations or their chemical structure. An example for methionine enkephalin, a peptide that contains 5 amino acids, is shown in the manual. 10 of the 20 amino acids are called essential amino acids because humans and other mammals cannot synthesize them and they must be ingested in food. Some amino acids can be synthesized in sufficient amounts in adult animals but are considered essential for young growing animals and must be provided in the diet. Specific to cats and ferrets is a requirement for taurine, which is often thought of as an amino acid but actually differs by the absence of a carboxyl, and then protein is broken down in the digestive system by gastric acid and the enzyme pepsin in the stomach. Gastric acid activates pepsin and begins denaturing proteins to allow further protein breakdown by pepsin. Other digestive enzymes such as trypsin, chymotrypsin, and elastase act further to hydrolyze proteins into units as small as individual amino acids ready for absorption through the wall of the small intestine. Amino acids are delivered to the cells via the bloodstream and distributed to tissues throughout the body. Once in the tissue cells, the amino acids can be used in protein biosynthesis as the building blocks for new structural proteins, enzymes, hormones. Vitamins are compounds that are essential in small amounts for the body to carry on its normal metabolic activity. Vitamins serve as chemical partners to enzymes in a variety of cellular reactions. Vitamins have chemical names, though they are often known by a capital letter, and some also have a number, like vitamin B1. Vitamins have diverse structures, with some being water-soluble, such as vitamin B and C, and others are lipid-soluble, such as vitamins A, D, E, and K. Vitamin C is ascorbic acid, a fairly simple organic acid, while vitamin B12 has a complex structure that contains a cobalt ion at its core. Some vitamins, such as D, K, and the B vitamin biotin, can be synthesized by the body. Others must be obtained from the diet. Although many animal species can synthesize vitamin C, other species, such as primates and guinea pigs, require a source of vitamin C in their diet. Primates and guinea pigs also require a source of vitamin C in their diet. Historically, vitamin C was very unstable, leading to shortened shelf life of these diets. However, newer feeds containing vitamin C use a stabilized form, which is L-ascorbyl polyphosphate, which increases shelf life. One should always consult the feed manufacturer regarding appropriate minerals are an essential part of the diet of all laboratory animals. These dietary minerals fall into two categories based on their biological importance: macrominerals and microminerals, respectively. There are 7 macrominerals. These are calcium, phosphorus, magnesium, sodium, potassium, chlorine, and sulfur. Calcium, phosphorus, and magnesium are required as structural components of the skeleton. Magnesium is also required for the activation of several enzymes. Sodium and potassium are important for maintaining isotonicity in body fluids and have a role in the electrical activity of cells. Sulfur is a component of several amino acids. All of the macrominerals are vital to the diet of laboratory animals. Their deficiency can lead to serious health problems, such as bone disorders, convulsions, anorexia, growth retardation, and death. The microminerals that are required for health in laboratory animals are more numerous, and their functions are more diverse. Many are components in enzymes and other proteins. For example, iodine in the hormones produced by the thyroid gland, and zinc in ribonuclease, DNA polymerase, and carbonic anhydrase. Microminerals may also interact with each other. For example, iron and copper are involved in the regulation of each other, switching reduced and oxidized states to maintain homeostasis. Note that while deficiency may have negative consequences, many trace minerals can have a toxic effect if supplied at too high a dose. A list of microminerals, their function, and the consequences of their deficiency are found in Table 8.1 in the manual. Fiber, sometimes referred to as roughage, is a form of carbohydrate which is indigestible to monogastrics and provides bulk for the digestive tract contents. These indigestible carbohydrates are usually plant components cellulose and hemicellulose. This fiber may either form a gel, such as psyllium husks, or remain entirely indigestible and fibrous, like cellulose. Another fiber component of plants is lignin, which gives plants a fibrous or woody character and is wholly indigestible. Fiber made of cellulose and hemicellulose is not broken down by mammalian enzymes, but can be broken down in the gastrointestinal tract through fermentation by bacteria. This process creates short-chain fatty acids that the animal can then use for energy or other biological processes. Ruminants can degrade fiber via the help of the rumen microbiome. Fiber degradation early in the gastrointestinal tract allows fiber to be the main source of energy for such animals. Monogastrics, however, have limited fiber degradation, which occurs much later in the gastrointestinal tract, which makes much of the energy from fiber metabolically unavailable. This occurs in the cecum and colon, depending on species. These species are often referred to as cecal digesters. These animals have a highly developed cecum and colon to handle fermentation. Examples are horses, swine, rabbits, and rodents. Certain species, such as rabbits, can benefit from coprophagy, the act of eating fecal matter to obtain lost nutrients, to recover some level of the fiber degraded can cause physiological effects such as decreased mineral absorption or alterations in gastrointestinal transit time and/or consistency of gastrointestinal contents. These effects may be beneficial to the animal's digestion and health. It's important that when examining the fiber content of lab animal diets, to be aware of the fiber requirements of each species under your care and to feed appropriately. Rabbits need higher dietary fiber than rodents, especially for general maintenance, usually a range of 18 to 25%. High fiber content in the diet reduces obesity in caged rabbits, promotes better digestion as bulk, and is less likely to cause digestive upset and inappetence. Feeding alfalfa or other hays is helpful to promote gut motility. Likewise, sheep need hay in their diet for normal rumen function postoperatively. Be aware that hay can plug drains requirements in indoor facilities. The animal's diet is a critical part of animal studies. A normal laboratory diet is intended to provide all of the required nutrients in order to provide a complete, nutritionally sound diet. Because a change in a normal diet can affect the animal in many ways, ensuring the appropriate diet will avoid adding a variable in the research protocol by inducing a physical change in the animal's health due to nutritional deficiency or excess. A laboratory animal's diet should be considered an experimental variable that affects research outcomes. It is a controllable variable because the diet can be altered by the researcher as required for an individual study. Facility staff should encourage researchers to use diets from reputable feed manufacturers who monitor ingredient and nutrient levels in diets over time by using an acceptable formulation system. In the lab animal industry, 2 diet formulations are considered acceptable. Fixed formulation means that the combination of ingredients is fixed such that every batch of formula made will have the exact same level of each ingredient as the previous lot. These ingredients are collectively referred to as ingredient inclusion, and the amounts of these are called ingredient inclusion levels. A fixed formulation may not account for slight differences in nutrients, as inherent variation will occur in ingredients in different crop yields. For example, consider a diet in which the amount of corn is fixed. As corn crops can vary in protein levels from year to year, the diet's protein content may also fluctuate slightly unless compensated by changes in other ingredients. The second type of formulation, called managed formulation, accounts for this natural ingredient variation by allowing small differences in ingredient inclusion from one batch of diet to another. The goal of a managed formulation is to maintain the same level of nutrition, such as protein, fat, etc., over time. Neither fixed nor managed formulations are affected by the market price of ingredients. A third type of formulation, called least cost formulation, relies on the market price of ingredients and is designed to provide the least expensive diet without accounting for how such changes may impact research. Reputable laboratory animal feed manufacturers never use least-cost formulations. Personnel should always be mindful of what the laboratory animals requirements are being fed. Many species have specific nutritional requirements. Most of the time, these needs are met by the appropriate feed provided. For example, both ferrets and cats are obligate carnivores and require a diet high in animal fats and proteins, yet low in fiber. Their diets must be supplemented with the organic acid taurine, which neither ferrets nor cats can synthesize. Reduced gluten diets may be appropriate for some species, as gluten, which is a polypeptide containing starch and lipid, has been implicated in enteropathy in non-human primates. The requirement of guinea pigs and non-human primates for vitamin C was mentioned previously. Genetic differences can also affect the nutritional requirements within a species. Age and physiological requirements can affect the amount and type of diet offered. Pregnant and lactating animals have higher nutritional requirements for fetal growth and milk production. For example, they require more protein and calories. In contrast, older animals may need fewer calories and less protein than young and growing animals. The content of laboratory animal feed may have effects beyond providing nutrition, and this must be considered when selecting the appropriate feed for a specific protocol. For example, chlorophyll in rabbit and rodent food can affect imaging studies by causing nonspecific fluorescence. Diets with reduced chlorophyll are available and should be used in imaging studies to reduce interference with study objectives. Soy protein has been shown to contain a class of phytoestrogens, which include isoflavones. Isoflavones are the most common form of phytoestrogens and can be measured in animal diets. Isoflavones mimic estrogen and can compete for estrogen receptors. They can affect the endocrine and reproductive systems of laboratory animals, altering breeding efficiency either positively or negatively. Furthermore, they can lead to either up or down regulation of estrogen activity in the body, which can have the side effect of inducing tumor growth in estrogenic tumors, inhibiting the action of tamoxifen, and attenuating the immune response. Animals on estrogen-sensitive studies should be fed a diet low in isoflavones. It is important to note that isoflavones and other phytoestrogens can also be found in many environmental objects mice come in contact with, such as water, water bottles, cages, and bedding. The potential for exposing animals to these chemicals via the diet and caging equipment should be considered when designing an estrogen-sensitive study. Special diets may also be required to study the effect of a particular nutrient, such as a sodium-modified diet to study the effects of sodium on blood pressure. The procedures conducted for a specific protocol may produce anorexia or increase nutritional requirements such as energy expenditure. Protocols may also restrict food or water intake. In addition to nutritional formulations, alternative forms of diet such as liquid or gel may be required during post-surgical periods. To procure a special diet, it is important to work with a feed vendor to select or develop a diet that meets the research requirement, for example, high sodium content, and is comparable with the standard diet fed to control animals. At times, there may be special diets to support research with other than nutritional needs. For example, tetracycline is included in diets to control gene expression in some genetically modified strains. Also, fenbendazole is often incorporated in the diet for treatment of internal animal models of disease. There are many ways in which biochemistry contributes to advancing the knowledge of disease processes in animals and humans. An experimental diet can be used to study the physiological effects related to specific nutrients and is used as one method to express a phenotype in animal models. An example of this is the use of high-fat feed to induce obesity and its subsequent effects on infectious diseases, atherosclerosis, or other disease processes. Dietary carbohydrates are rapidly converted to blood glucose, and insulin is the principal hormone that regulates how much of the circulating glucose is taken up by most cells. Diabetes mellitus results from the defective secretion or action of insulin, which leads to hyperglycemia, or high blood sugar levels. Since glucose is unavailable to the cells of a diabetic, the body begins to break down fat as an energy source, and there is an overproduction of ketones as a result of increased fat metabolism. Overproduction of ketones can lead to a toxic condition called ketoacidosis that, if left untreated, can lead to diabetic coma or even death. Type 1 diabetes, or insulin-dependent diabetes mellitus, accounts for up to 10% of diabetes cases. Non-obese diabetic mice are used by researchers to identify IDDM susceptibility genes and to understand how the gene is expressed as well as the disease mechanisms involved in the human form of the disease. Lipids are an important nutrient, but abnormal lipid metabolism and deposition can be a serious problem. Atherosclerosis, in which the body's blood vessels become narrowed and cannot allow the normal volume of blood to circulate, is caused by cholesterol deposition on the inner lining of blood vessels. This deposit is called plaque. While plaque deposition is not entirely understood, it is known that an imbalance in the different types of lipoproteins, high density and low density, is a major contributing factor to this disease. High-density lipoproteins, or HDL, and low-density lipoproteins, or LDL, are two classes of lipoproteins that transport lipids through the blood and body fluids, and they aid tissue cells in taking up the lipids. Atherosclerosis is common in humans with increased levels of low-density lipoproteins, but occurs naturally in very few animal species, making it difficult to study the possible causes of this condition. The technique of genetic engineering has enabled scientists to create mice that overexpress cholesterol and other lipids thought to play a role in this disease. These mice develop atherosclerotic lesions, very similar to those in humans, and have helped advance the knowledge about this disease. Atherogenic diets are used to help induce and study cardiovascular diseases. Phenylketonuria is a genetic disorder for which human newborns may be tested. An enzyme called phenylalanine hydroxylase transforms the amino acid phenylalanine into tyrosine. This enzyme is lacking in individuals with phenylketonuria. Therefore, phenylalanine accumulates in the blood and body tissues, and this buildup can cause mental retardation. If phenylketonuria is detected early, a diet low in phenylalanine can prevent this problem. There are strains of mice that lack the phenylalanine hydroxylase gene. The symbol for this strain is PAH. Research using this animal model is helping scientists to elucidate this thorough understanding of the biochemical compounds that support living systems is essential for ensuring the health and well-being of laboratory animals. As these compounds are all important components of animals' feed, the diet has a fundamental influence on health. Nutritional imbalance, either deficiency or excess, can cause disease. By ensuring laboratory animals receive complete and appropriate nutrition, technologists maintain the integrity of the research, so the data generated are accurate. Molecular biology. Molecular biology is the study of the formation, structure, and function of the biologically important macromolecules that are found within living cells. This includes those molecules that involve the genes and the products of those genes. In this chapter, you'll learn about deoxyribonucleic acid, DNA, ribonucleic acid, RNA, and protein synthesis and how they are related. In eukaryotic cells, the nucleus is the storage site for genetic information, and DNA is the macromolecule that contains it. In the nucleus, the DNA is condensed into chromosomes. The number of chromosomes varies among species. Figure 9.1 in the manual illustrates how DNA is organized in the nucleus into chromosomes and how genes are segments of these chromosomes. Understanding this cellular machinery has enabled scientists to understand how the inherited characteristics that we observe every day, for example, eye color and body size, are linked to the underlying chemical processes that produce them. Chromosomes are arranged in pairs in the body's cells, with the exception of the germ cells, also called gametes. Based on the number of chromosomes present, cells are often categorized in two ways. As diploid cells, these cells have chromosomes that are paired. Diploid cells are commonly known as somatic cells and make up all tissues throughout the body. These cells have single chromosomes that are not paired. The only haploid cells are the ovum, which are found in the ovaries, and sperm, which are found in testes. Table 9.1 in the manual shows the number of chromosomes in diploid and haploid cells of different species. Chromosomes are made up of large numbers of genes. A gene is a sequence of DNA that codes for a protein. Each gene is a unit of information, and it occupies a very specific location on the chromosome. Genes can be thought of as the blueprints upon which all the cell's activity is based. The characteristics of an organism are in good part determined by the sum of the effects of all of its genes, and genes direct the chemical reactions occurring within each organism. The number of genes is estimated to be close to 30,000 in both human and mouse diploid cells. The genetic makeup of an organism, the sum of all its genes, is called its genotype. The phenotype of an organism represents its actual physical or behavioral characteristics such as height, weight, hair color, and so on. The phenotype is determined by the animal's genes but is also influenced by the environment. Identical twins, for example, have the same genotype, but their family and close friends can usually tell them apart because of subtle differences in their phenotypes. A permanent change in the DNA sequence of a gene is called a mutation. A mutation can occur spontaneously or be caused by exposure to certain chemicals or types of radiation. A mutation that occurs in the germ cells of the animal, the ovum or sperm, will be transmitted to the next generation. If a mutation occurs in other cells, it only affects that animal and is not transmitted to its offspring. Scientists can now manipulate the DNA of cells to create mutations. This methodology, called genetic engineering, will be discussed in a later chapter in this manual. Deoxyribonucleic acid, DNA, is a polymer, which means that it is composed of a series of chemical subunits linked together. These chemical subunits are called nucleotides. In DNA, each nucleotide consists of a sugar, deoxyribose, a nitrogen-containing base attached to the sugar, and a phosphate group. There are 4 kinds of nucleotides in DNA, which differ from each other by their nitrogenous base. The 4 bases are adenine, A, guanine, G, cytosine, C, and thymine, T. The arrangement of these 4 bases along the DNA molecule determines the genetic makeup of the organism. The DNA molecule is best described as a double helix, which is similar to a twisted ladder. The sides of the ladder consist of the sugar of one nucleotide attached to the phosphate group of the next nucleotide. This arrangement repeats itself all along the DNA strand. This arrangement of phosphate group, then a sugar, another phosphate group, then another sugar, is referred to as the backbone of DNA. The nitrogenous bases form the rungs of the ladder, with the base of the nucleotide on one strand forming a bond with the base of the nucleotide of the other strand. These bases pair with each other in a specific way referred to as complementary pairing. Adenine will bond only with thymine, and cytosine will bond only with guanine. The phosphate sugar bonds in the backbone are strong bonds. However, bases are held together by weaker hydrogen bonding. This type of bond will be shown to be important later when replication is described. Figure 9.2 in the manual illustrates how the nucleotides are arranged to form the backbone of each DNA strand and how the bases form bonds that hold the 2 strands together. Ribonucleic acid, RNA, is similar in structure to DNA. It is composed of a long polymer of a simple sugar attached to a phosphate group and to a nitrogenous base. The sugar in RNA is ribose rather than the deoxyribose found in DNA. 3 of the 4 bases in RNA are identical to those in DNA, but the 4th RNA base is uracil. Uracil takes the place of the thymine found in DNA. Another difference is that RNA is usually single-stranded, maintaining its structure by folding over upon itself. RNA is vital in eukaryotic cells as a component of protein production machinery. The process of protein synthesis is discussed later in this chapter. RNA may also be the sole nucleic acid used by the organism. Many viruses have either a single or double-stranded RNA genome instead of using DNA to encode their genes. Cells reproduce by dividing. A parent cell divides into 2 daughter cells. In order for each daughter cell to have the same genetic makeup as the parent cell, the DNA in the chromosomes must be duplicated prior to cell division. This process is called replication. The first step in duplicating the chromosomes requires that the 2 strands of the DNA double helix separate. The separation occurs at the hydrogen bonding sites of the base pairs. As mentioned earlier, these hydrogen bonds are weak, so they are easily broken. Once the two strands are separated, the bases of free nucleotides within the cell nucleus begin to bond to the single DNA strand. Just like on the original strand, adenine-thymine and guanine-cytosine are the only possible bonds between the bases. Once all of the bonds have been completed, two exact copies of the original chromosome have been produced, each one containing one of the original strands and a newly formed strand. Replication is illustrated in Figure 9.3 in the manual. When the cell divides, each daughter cell gets an exact copy of the original chromosomes and is diploid like the parent. Mitosis is the process in which a eukaryotic cell nucleus splits in two identical cells, followed by division of the parent cell into two daughter cells. The major purpose of mitosis is for growth and to replace worn-out cells. If not corrected in time, mistakes made during mitosis can result in changes in the DNA that can potentially lead to genetic disorders. The word mitosis means threads, and it refers to the thread-like appearance of chromosomes as the cell prepares to divide. The chromosomes replicate as discussed previously and line up as pairs along the center of the dividing cell. The paired chromosomes then pull to either side of the cell, and the cell splits into two daughter cells. In the process of sexual reproduction, diploid cells in the ovaries and testes undergo a form of cell division. This process is called meiosis, and it results in haploid ova and spermatids. The ovum and spermatids each contain half of the chromosomes found in the parent cell, thus the term haploid. The process of meiosis is similar to mitosis, but there are two crucial differences. First, during meiosis, when the chromosomes replicate before division and line up at the center of the cell, the chromosomes recombine, switching alleles on the chromosomes and slightly changing the genes so the haploid daughter cells are all slightly different. Secondly, meiotic cells undertake a second division, which divides the chromosomes to create four haploid daughter cells, each containing a mix of the parental genes. When an ovum or egg and a sperm unite, a zygote is produced. The zygote is genetically the offspring of both parents. The zygote has paired chromosomes. One set is from the mother and one set is from the father, so the zygote is a diploid cell like the parents. The processes of meiosis and mitosis are shown in Figure 9.4 in the manual. There are two steps that allow cells to read genes and produce amino acids: transcription and translation. The first step of gene expression is transcription, in which RNA is made from a DNA template. The second step is translation, which is the process of messenger RNA, or mRNA, being translated into amino acids of a protein. This is protein synthesis. Transcription. The DNA contains all the information that directs protein synthesis, which in turn controls cellular metabolism. However, the genetic material is kept in the nucleus, and the protein manufacturing machinery is in the cytoplasm. In order for the cell to read the genetic code to make protein, a process is needed by which information is transported to the site where the reactions occur. Transcription is an essential step in that process. It is the synthesis of a messenger between the two sites. Transcription begins in a manner similar to replication, in which the two strands of DNA separate at the base pair binding sites. On one DNA strand, complementary pairing takes place with free base pairs. However, in transcription, it is ribose-containing nucleotides that form the complementary binding, which is guided by the RNA polymerase. The result is a new strand of RNA. This strand of messenger RNA, mRNA, contains the complementary information from the single strand of DNA from which it formed. Figure 9.6 in the manual illustrates transcription. The mRNA crosses the nuclear membrane and into the cytoplasm. This mRNA is the bearer of instructions from the DNA to the cell's protein-synthesizing machinery located in the cytoplasm. Translation is the cellular process by which the genetic information in the mRNA, which is now in the cytoplasm, is read and used to synthesize proteins and related compounds. In the cytoplasm, the steps for protein biosynthesis are: 1. The mRNA attaches to a ribosome, the protein manufacturing machinery of the cell. Sequences of nucleotides in the mRNA code for specific amino acids. 3 base pairs code for an amino acid. 2. The ribosome moves along the RNA until it reaches a start codon, the sequence of nucleotides that will initiate translation. Transfer RNA, or tRNA, molecules, each carrying a newly synthesized amino acid, move into the ribosome. The tRNA anticodon binds to the mRNA. 3. As more tRNAs move into the ribosome, the amino acids bind to each other and the tRNAs are released, beginning the amino acid chain that makes up a protein. The order of the amino acids and how they interact provide the protein structure. And 4. Once the ribosome reaches a stop codon, the amino acid chain is released and will either be used or undergo post-translational modification. Figure 9.7 in the manual illustrates the process of translation by which proteins are synthesized post-translational modification. For example, proteins are often synthesized as inactive precursors that must undergo additional steps before they become active and can fulfill their role in the cell. These modifications may involve removal of some amino acids from the end of the protein, the addition of lipids or sugars, or a structural change that alters the shape of the protein. In summary, chromosomes contain DNA, which is the genetic material that directs all functions of the cell. Every time a cell divides, its DNA replicates so that the daughter cells are clones to the same genetic makeup as the parent cell. See mitosis. To synthesize cell proteins and related compounds, the double-stranded DNA in the nucleus is transcribed to form a single-stranded messenger RNA, which migrates to the cytoplasm where it is translated by ribosomal and transfer RNA in the synthesis of proteins. The information contained in the mRNA sequence of nucleotides is used by the ribosomes to connect amino acids in a chain forming a protein. Depending on the particular protein, additional steps may occur to complete the final molecular biology. Understanding how DNA functions has enabled scientists not only to study the functions of genes, but also to artificially manipulate genes to create animals and plants with specific genetic traits. Traditionally, this has been done by breeding generations of parents with the desired trait, but it can now be done by constructing the desired genetic code in a laboratory and inserting this code into a zygote or embryo during development. This process is called genetic engineering and will be discussed in a later chapter. In order to understand how genes are identified and manipulated in the laboratory, it's necessary to understand several techniques. These techniques include collecting the DNA, purifying it, cutting it into smaller fragments, separating the fragments, making large amounts of the desired fragments, and testing to be sure the isolated fragment contains the desired gene. The gene can then be inserted into an animal to create a specific purifying DNA. Collecting DNA involves chemically breaking open some cells to release their contents, including protein and DNA, into solution. The DNA is purified through a series of steps of dissolution and centrifugation. In the final step, DNA is collected as a pellet in the bottom of a centrifuge tube. The genetic material isolated from cells is composed of very large pieces of DNA containing perhaps thousands of genes. Most scientists are interested in only 1 or 2 genes, so the DNA then has to be cut into smaller pieces. This is accomplished by adding special enzymes called restriction enzymes to the DNA sample. These enzymes bind to DNA at specific base pair sequences and cut the DNA at those sites. Different restriction enzymes bind at different sites, so adding several different enzymes to a sample will result in the DNA being cut into many fragments. Pieces. The sample now contains a mixture of DNA pieces of different lengths, that is, containing different numbers of base pairs that are separated using a technique called electrophoresis. In electrophoresis, DNA is applied onto a gel to which an electric current is applied. The DNA fragments migrate and separate according to their size and electrical charge. A stain is used to visualize the DNA fragments, which show as bands of colored material on the gel. The fragment of interest can then be isolated from the gel by cutting the specific fragment. This fragment is then placed in solutions to dissolve the gel. Figure 9.8 in the manual shows an example of a gel with DNA fragments. Cloning is using specialized DNA technology to produce multiple exact copies of a single gene or other segment of DNA to obtain enough material for further study. When extracting and purifying DNA, sometimes only a very small amount of the DNA fragment can be recovered, so cloning techniques are helpful for increasing the amount of the genetic material for use in research studies. In the laboratory, cloning is accomplished by either using bacteria or a process called the polymerase chain reaction, or PCR. In the bacterial technique, a DNA fragment of interest may be incorporated into bacterial cells, such as E. coli, by exposing them to an electric field, a technique known as electroporation. This allows the DNA to pass through the cell membrane and incorporate into the bacterial genome. These bacterial cells are then grown in culture, making millions more cells containing the piece of desired DNA. The DNA is then extracted from the bacteria using the methods polymerase chain reaction, PCR, is another amplification technique for producing large quantities of a DNA fragment of interest. To test an animal's DNA using PCR, a sample of tissue is processed to extract the DNA from the tissue cells. In mice, this sample may be obtained from the tip of the tail or with less invasive sampling methods such as plucking a hair shaft from the skin, taking a swab of cheek cells, using the tissue from ear punch identification, or collecting fecal pellets. The tissue is dissolved and a mixture is prepared in a tube containing the following: the tissue DNA, known as the template, an enzyme known as a polymerase, specially designed short DNA fragments known as primers that match the gene of interest, and nucleotides that are the building blocks of a new DNA strand. This tube is placed in a PCR machine. In PCR, double DNA strands are first separated by exposure to a high temperature. A polymerase enzyme synthesizes new strands of DNA complementary to the target DNA sequence. This enzyme replicates the DNA through many cycles, which is a process called amplification. Each cycle doubles the amount of DNA fragments. 20 cycles of amplification can increase the amount of original DNA fragment 1,000,000-fold. A standard PCR can be used to detect the presence of a targeted DNA sequence. Of biomolecules. Detecting nucleic acids, quantitative RT-PCR. When a gene is expressed, mRNA is produced to transmit the information from the genetic material to the protein-making machinery of the cell. Quantitative PCR amplifies these mRNA transcripts and incorporates a fluorophore, allowing differences in the relative abundance of mRNA transcript numbers to be measured. Specific gene expression levels match the strength of the fluorescent signal, which is measured relative to the expression of housekeeping genes. These are genes related to cell structure or metabolism that are constantly expressed as a product of cell survival and function. The benefit of qPCR compared to other RNA detection techniques is that it is quantitative. Other techniques, such as blotting, can only indicate a trend in abundance as measured by the strength of the band that is produced on the radiograph. Blotting also requires a great deal more RNA than qPCR to see a useful result. Both of these factors have led to the decline of blotting compared to qPCR as a diagnostic technique. Because qPCR only needs a tiny amount of RNA to produce a meaningful measurement, it has made other diagnostic techniques viable. Environmental testing, such as the swabbing of air filters and cage rack plenums, is now possible using this technique. Unlike more invasive or expensive techniques, such as the necropsy and sampling of sentinel mice, the use of a qPCR analysis on environmental samples allows for the quantitative detection of pathogens quickly and easily. These techniques and their use is covered in greater detail in Chapter 6, Health Surveillance and Quality Assurance. Western blot. To detect protein, the most commonly used method is the Western blot. Samples are separated by gel electrophoresis and then transferred to a flexible membrane. This membrane is incubated with an antibody specific to the protein of interest, then washed and exposed to a secondary antibody that is linked to a peroxidase enzyme. When the membrane is subsequently exposed to a chemiluminescent substrate, the protein bands on the membrane produce light and can be visualized using photographic film. Western blots are often used as a secondary method of genotyping transgenic and knockout animals, as the Western blot will show if genetic deletion has deleted the expression of the target protein as well. The process for Western blotting is shown in Figure 9.9 in the manual. An understanding of the principles presented in this chapter is necessary for beginning to understand one of the most incredible and important scientific advances in history. The techniques of molecular biology have allowed scientists to manipulate the genetic code of an organism, in effect, to genetically engineer an organism. Genetic engineering is destined to be the main focus of scientists in many fields of biomedical research for years to come. The next chapter, Genetic Engineering, discusses some of the techniques and results of this exciting field of research. Genetic engineering. Genetic engineering refers to manipulation of genetic material using chemical or biological means, whether in a test tube, which is called in vitro, or in a living organism, which is called in vivo. Biotechnology has allowed pieces of nucleic acid, either DNA or RNA, to be moved, changed, and reassembled, all with the goal of better understanding the basic functioning of genes. The work of genetic engineering involves many individuals, including the investigator, the research technician, and the animal technician. The first step is creating the DNA construct, which is an artificially created segment of nucleic acid that is usually created by the transgenic facility or a commercial vendor. This DNA construct is then placed in an animal by a scientist trained in this field. To generate transgenic animals successfully, each step is critical, from designing the DNA construct to embryo transfer. The animal is bred and evaluated to ensure the genetic change is passed to offspring and expressed as a desired phenotype of physical or behavioral trait. The investigators and the animal facility staff work together to manage these breeding colonies. This includes phenotyping animals to assess physical or behavioral traits and analyze the results, and providing animal husbandry. Laboratory animal technologists should understand the basics of this science because frequent advances in this field make an impact on the number of animals produced, the housing and husbandry needs, and therefore the operation of the animal laboratory animals. Animals are good models for studying diseases that affect humans because much of the genome is conserved across species. The mouse in particular has become the premier animal model for biological conditions. For example, about 40% of the human and mouse genomes can be directly aligned with each other, and about 80% of human genes have a corresponding gene in the mouse genome. The methodologies to manipulate the mouse genome have grown extensively since the first transgenic mice were created in the early 1980s. The exponential growth of new mouse models has presented many new challenges to laboratory animal personnel. Some laboratory animal facilities are now totally dedicated to the housing and husbandry of genetically engineered mice. In addition to being the easiest mammal to genetically engineer, the mouse is a popular research tool because of its small size and consequent ease of housing, short gestation period, and the availability of genetically defined inbred strains from commercial suppliers. Many of the basic principles of genetics and breeding that will be discussed here are outlined in Chapter 8, Genetics and Breeding, in the Laboratory Animal Technician Training Manual. A review of that manual's chapter may be helpful at this time. This chapter will focus on the laboratory mouse, but the genetic engineering techniques described for the mouse can also be used, with minor modifications, for other species, such as frogs of the species Xenopus laevis and zebrafish, which are the species Danio ririo. Because the genomes of many plant and animal species, including humans, have been entirely sequenced, the DNA structure is readily available. The development of this genetic library has facilitated the genetic manipulation of these species as well as other laboratory animal species, including rats, swine, sheep, Table and 10.1 in the manual. Genetically modified animals are critical for understanding the biochemical and physiological processes of disease. In addition, by assessing the genes involved in development, we can better understand how birth defects are caused and maybe mouse models. There are two general categories of mice that are generated using genetic engineering techniques. One is transgenic and the other is gene-targeted. Transgenic mice are produced by adding a gene from one animal into another. Gene-targeted mice are produced by altering a gene that is already in the animal. Each category is used to answer different questions. Transgenesis generally refers to introducing additional DNA into the genome of an animal. Trans means across or to move, as in transport. The DNA may originate from the same or another species. The basic steps in genetic engineering involve preparing a DNA fragment and inserting it into the nucleus of a cell where it integrates into the cell's DNA. The fragment of DNA that is transferred is called the transgene. Transgenic organisms have a fragment of DNA that has been introduced into their genome. There are three primary ways in which genetic engineering techniques are used to generate transgenic mice. One is by microinjection of the transgenic construct into a fertilized egg, which is called an oocyte or zygote. An alternative way is to introduce a transgene by use of a retroviral vector into the egg. Finally, the transgenic construct can be introduced into embryonic stem cells, and then the embryonic stem cells are injected into mouse blastocysts. Transgenic animals are valuable in that a gene can be produced in the laboratory that meets a specific need of the investigator. Some examples of the types of research conducted with transgenic models include toxicology for the detection of toxic agents, oncology, which involves creation of mice that can accept human tissues, and genetically engineered hormones, managing a transgenic facility. The production of genetically engineered animals involves high costs and may create an immunodeficient animal or animals requiring unique supportive care needs. Extensive precautions are required to protect the investment of their creation. Thus, the LATG working in a transgenic facility needs to have an in-depth understanding of the goals of each animal colony, the potential for biological contaminants or pathogens, and the timing involved with production of eggs, embryos, and pups. Quality assurance of a transgenic colony through genetic analysis of tissue samples may become one of the responsibilities of the LATG. In addition, the LATG may also need to keep the investigator and staff informed of mating events, breeding successes, animals ready for weaning, and other important issues. Time management and priority setting are discussed elsewhere in this manual and are especially important skills for the LATG working with genetically modified mouse animals. Breeding rooms must also be determined for all members of the research team. Both husbandry and research personnel need to understand the special housing and colony health surveillance testing needs based on the unique genetics of the animal. These special husbandry needs may result in different access designations required to protect the animal. Examples of the standard access types include 1. Full access, which increases the risk of introducing disease entities to valuable genetically modified lines. 2. Limited access, which allows PIs to conduct some manipulations, including moving animals in and out of protected caging, but with facility personnel having the majority of contact with the animals. And 3. Barrier, where investigators' access is restricted, so they must rely on the animal care staff to handle all in a transgenic animal facility. A great deal of time and effort is needed to create transgenic mice. The whole process is dependent on good husbandry practices and alert technicians. Successful matings must take place routinely to produce the fertilized eggs and blastocysts for injections and to provide the pseudo-pregnant foster mothers into which the embryos are surgically transferred. Finally, once pups are born and weaned, they must be genotyped and then bred appropriately. In this section, we'll look at what must be done within a transgenic facility. The LATG in a transgenic animal facility may be expected to check females for estrus, set up matings, check plugs after mating, administer hormone injections, monitor health status of implanted females, examine gravid females for pups and then monitor the pups for phenotype, maintain fertile colonies of males, and record plugging behavior of males. The process of making genetically modified mice usually results in the production of many mice that may eventually not be needed. Control over numbers is a concern from a labor and management perspective, as well as IACUC oversight and ethical concerns. Initially, it may be necessary to prove that the gene of interest has truly become incorporated in a newly engineered line. Thus, many animals may be housed until the genetic makeup is characterized. All animals are usually sampled for tissue to be used in genotyping. Then the animals of value to the research study are kept and the remainder euthanized. For an LATG working as a supervisor, this can mean wide variations in labor assignments because caging, animal care, and technical duties will fluctuate. Accurate records must be kept, and paperwork can consume considerable colonies of genetically engineered mice. LATGs may provide care for transgenic and targeted mice outside of the transgenic animal facility once animals are transferred to the investigator and moved into the general mouse population. At this point, colony health maintenance will require attention to mouse breeding and the attendant problems with breeding, as well as different phenotypes that may manifest. Each facility will have a mechanism for reporting health concerns that will guide you on how to observe and document these breeding and unique transgenic animals. It can be argued that genetically modified animals are just like any other in the facility. They must be housed, observed, fed, watered, and managed as all other mice. Indeed, in many cases, a transgenic or gene-targeted strain may not have a noticeably different phenotype, and to the casual eye may appear similar to its wild-type counterparts. Still, there may be some special needs that must be addressed by staff involved in the care of these animals. This section will highlight special considerations when it comes to the care of genetically engineered mice. There are many factors that make the creation of genetically engineered pups difficult. The embryos may not successfully implant in the uterine wall or may die in utero. The pups may die during or shortly after birth, or they may be cannibalized by one of the parents. The progress of gestation should be monitored in order to predict the timing of delivery, which is called parturition. The telltale sign of a pregnant mouse is the bulge of her abdomen to the point where she looks pear-shaped. This may not be as apparent if she's carrying a very small litter. If she no longer appears pregnant and pups are not neatly nested in the cage, the vulva should be examined for evidence of birthing or dystocia and bedding searched for remnants of pups. Any tissue can be collected and sometimes analyzed for the presence of the gene or chromosomal abnormalities. By the same token, a timed mating that fails to result in embryos or pups may be evidence of sterility or embryonic lethality due to the genetic modification. Record keeping for breeding colony maintenance may be standardized at your facility or be unique for each laboratory. Assuming a litter has been born successfully, it's important to note the exact date of birth and, after 5 to 7 days, count and record the number of pups. At that time, you can also check for the presence of a milk spot, and, knowing how many pups were born, account for the loss of pups later on. One should observe the condition of the nest and whether the pups are grouped together, which is a sign of good maternal care. Pups scattered about the cage may be a sign of maternal neglect. The dam should spend most of her time lying on top of the pups. All of these observations provide a measure of the mothering instincts of the dam and of the health of the pups. Be aware, though, that even good dams may not take care of sickly pups. The loss of pups should be reported to the research staff, as it may be an indication of the gene's effect on viability or its effect on mothering instincts. The lack of a milk spot may suggest some problems with lactation, and nutritional intervention may be needed. The pups could be fostered to another nursing mother, or the technologist may need to provide a milk substitute. Of course, any action taken by the husbandry team will depend on the institution's animal care program, the protocol, and SOPs. While these observations are necessary, it is important to disturb the mother as little as possible, since this can increase the incidence of cannibalism, particularly in C57BL6 mice. Thus, the use of clear nesting boxes and the ability to look through the cage to examine pups can be valuable. Looking through the cage bottom may give you a view of the pups in the bedding. The technician should watch the pups grow and look for potential differences in typical age markers, such as eyes opening and fur coming in, runted pups, and readiness for weaning at the typical time. Animal health concerns. The technologist must closely observe the animals in his or her care and train others to be just as attentive. In particular, the guide states that the first generation of a newly created line of genetically modified animals should be observed carefully for signs of pain, distress, or disease, as with all animals. Problems may develop over time, so animals should be observed carefully on a regular schedule as determined by your facility SOPs. A gene's phenotype may manifest at different times, including in utero, for example, causing embryonic death and a smaller litter, as the offspring grow, such as a runted phenotype, or during adulthood, for example, a shortened lifespan or a defective anatomical structure. Another possibility is that a breeding pair may not reproduce as expected because one or the other has a reproductive dysfunction. Reporting these observations to the research staff is extremely important. Table 10.2 in the manual lists some of the more common health problems found in genetically modified animals has no guarantee of success. There may have been a misidentification during the sexing of the animals. Some strains have a higher probability of sterile males, and some females do not develop maternal instincts or will cannibalize their young if overly stressed before a technician is even aware that the offspring have been born. Dystocia can also be a problem, and oxytocin used to induce labor may not be effective. Females may not even appear pregnant due to the small number of embryos, some of which may have died in utero due to a genetic lethality. All mice, whether genetically modified or not, must be observed for routine health problems. While clinical signs of illness may occur in any mouse, encountering them in a genetically modified mouse may require a different response, as this mouse could possibly be the only one in existence. Thus, while one might routinely euthanize a wild-type mouse with such a problem, it may be important to save either the mouse using extraordinary measures, or if the mouse is moribund, at least to save the gonads so they can then be used to generate pups. If a transgenic mouse is found moribund, it is helpful to contact the transgenic facility to determine if they can preserve the ovary if a female or sperm if a male. Staff awareness of and training on the special needs of genetically modified animals are essential to ensure human care of these valuable animals. There are times when large numbers of animals are not used in a study and must be euthanized. For the transgenic animal facility, fulfilling the principles of the 3 R's can become a concern. In many cases, the colony must be overbred to obtain the number of mice needed with a particular genotype, and the remaining animals generated must be euthanized. The animal care staff may also need the support of the supervisor or manager to deal with their emotional feelings when large numbers of animals are scheduled for euthanasia. It can be difficult to work weeks or months building a colony only to euthanize a large number of animals because the gene expression is not adequate. Transporting genetically engineered animals. Because genetically modified animals have become so important as research models, there is an active exchange of mice between institutions. Mice may be created in a central transgenic facility and then shipped to various laboratories nationwide and worldwide. This has led to 1. The creation of specialized import-export staff positions, sometimes known as shipping coordinators, for oversight of animal exchanges. 2. The need for frequent sentinel testing to review the health status of the mice due to the accidental introduction of a previously excluded microorganism, whether pathogenic or not. 3. The need for increased quarantine space to confirm that incoming mice truly do not carry unwanted microorganisms or pathogens. And 4. Extensive paperwork between institutions, including material transfer agreements, custom regulations, and intellectual property concerns, and the need for a better understanding of the laws and regulations for transport between countries. One method that is available that can override many of these concerns is to ship cryopreserved ovaries, embryos, or sperm. As these tissues can be used to regenerate the mouse line and generally cannot support the growth of pathogens, shipping of cryopreserved tissue is an excellent method of transporting mice easily and cleanly. The LATG should be aware of institutional capabilities for cryopreservation and recovery of germ tissue. The transport of animals in and out of the facility can also be a problem. Investigators may want to remove animals for study, such as behavioral testing or imaging, and then return them to the facility. Animals that have been taken out of the facility could pose a risk upon return from having picked up a microorganism. Innovative equipment that meets transport needs has been created, but sometimes other solutions must be developed. Therefore, a rodent health surveillance program must be developed to monitor the colony health based on transport of mice within an institution, as well as criteria for imports. Imported animals may require a period of quarantine prior to being released into the established animals. Colony. Types of animals needed. 4 groups of mice are needed for the production of transgenic animals, which we're about to describe. The groups are egg donor females, embryo recipient females, stud males, and vasectomized or sterile males. Egg donor females are those females that will be used to generate the eggs or embryos for the procedures used in the transgenic facility. Because many eggs or embryos are needed, a procedure called superovulation is often used to induce the release of many eggs or embryos for each female. To superovulate a female, each female mouse receives 2 intraperitoneal injections of exogenous hormones, pregnant mare serum gonadotropin, and human chorionic gonadotropin. Pregnant mare serum gonadotropin acts like follicle-stimulating hormone and is given first to physiologically prepare the female to ovulate. Between 46 to 48 hours after the pregnant mare serum gonadotropin injection, human chorionic gonadotropin is injected and causes the release of the ova. This hormone causes the release of 3 to 4 times as many eggs from their ovaries as the mouse would produce naturally in one estrus cycle. Superovulation allows many embryos to be harvested from each female, thus reducing the number of donor mice needed to create a genetically modified line. The increase in yield varies from strain to strain, with hybrid strains generally responding better than inbred strains. If fertilized eggs are required, Females are mated
Many
overnight
factors may
with
affect
stud
the
mating. Efficiency of superovulation, including the strain of mouse, the female's age, her weight, the timing of the hormone injections, and the light-dark cycle. The dosage and the time of day at which each hormone is injected may vary depending on the strain of mice and the light cycle of the room where they're housed. The time of day for injection must be closely followed in order to obtain the maximum number of ova. Typically, mice procured for superovulation should be allowed to adjust to the facility's light-dark cycle before receiving these injections. To check for successful mating, the females are examined for vaginal plugs the morning after mating. This helps monitor the performance of the stud males and can also indicate whether the hormone injections were administered correctly. A high percentage of females that have been correctly hormone treated should have a plug the morning after the human chorionic gonadotropin injection and the mating. The timing for harvesting the ova or fertilized eggs depends on the objective. If pronuclear injection of DNA is planned, then the fertilized eggs are harvested from the oviducts of euthanized females the morning after human chorionic gonadotropin injection and mating, or day 0.5. For cryopreserving embryos, either 2-cell or 8-cell stage embryos are generally used. 2-cell embryos are obtained by harvesting the day after human chorionic gonadotropin injection and mating, and then allowing the embryos to culture overnight, day 1.5. 8-cell embryos are harvested on the morning of the second day after mating, day 2.5. If blastocysts are desired, the embryos are harvested from the uterine horns in the morning of the third day after mating, day 3.5. If an in vitro fertilization is to be performed, the timing is completely altered to take into account the time at which the egg is most amenable to fertilization, typically between 12 and 17 hours after human chorionic gonadotropin injection. This means that hormone injections are given in the late afternoon or evening, from 2:30 p.m. to 7 p.m., depending on the strain of the female. The eggs are harvested early the following morning and placed into drops of swimming sperm for fertilization. This procedure is often used to generate many mice at the same time for cryopreservation or line expansion, or to rescue a male that no longer engages in mating behaviors. Once embryos have been obtained after injection or after isolation for other purposes, they must then be put into females capable of carrying a pregnancy. The embryos will implant in the uterus and develop into fetuses. The pseudo-pregnant females are known as recipients or surrogate mothers. These females undergo normal hormonal changes that prime their reproductive tracts to carry embryos to term, but their own eggs are not fertilized. Unlike the donors, these females are not superovulated. When bred with a vasectomized or sterile male, the recipient females become pseudo-pregnant simply because the physical act of copulation stimulates their reproductive system to prepare them to become pregnant. Their mating is synchronized to match the stage of the fertilized eggs they will receive as implants so as to physiologically meet the needs of the developing embryos. These embryos develop and pups are born at the end of a normal gestation period. The female is called a surrogate mother because the pups are not her own offspring, but rather the offspring of the donor female and the stud male. Embryos can be transferred to recipient mice either surgically, especially if embryos are less than 3.5 days, or non-surgically for embryos at 3.5 days. For surgical transfer, recipient mice are anesthetized and prepped for the surgery. For pronuclear injections, up to 15 1- or 2-cell embryos are transferred into the oviduct. In contrast, blastocysts injected with embryonic stem cells are usually transferred into the uterine horn using a similar surgery. Following the surgery, the mother must be monitored to ensure a good recovery. The gestation time for the mouse can vary by strain, but is typically 19 to 21 days. When the recipient females give birth, their pup's survival depends on good maternal instincts, which can be disrupted by poor husbandry practices and environmental disturbances or stressors. Some of the more common stressors include cage disturbances, loud and/or sudden noises, vibrations, lighting changes, both intensity and light cycle, and personnel changes. Construction activities taking place in or around the building are a common source of many of these stressors and should be coordinated and closely monitored. Lighting changes often occur due to research staff entering rooms during the dark phase and turning on white lights. During the dark phase, red lights are used as an alternative lighting system, and controls are implemented to prevent activation of white lights. It's important to minimize disturbances and handling of the cage from gestation day 15 to week 1 postpartum. Mothers and pups during gestation and neonatal development should be monitored for signs of problems and a need for intervention. It may be necessary to place the pups with another mother, called cross-fostering, to enhance their survivability because the mothers may neglect or cannibalize their young at any time until weaning. Weak offspring may need nutritional supplements. Some neonates may die, in which case the investigators may want to examine their tissues. It is therefore important that these animals be found quickly before their tissues autolyse. Fertile males of the same strain background are used for mating to superovulated females to produce eggs or blastocysts. It's important that highly fertile young males are used as stud males to ensure that all of the eggs are fertilized. Typically, stud males should be of maximal breeding age, 3 months to 8 months of age. Because mated males are often aggressive, stud males are housed singly. For maximal fertilization, only one superovulated female is introduced into the stud male cage after receiving human chorionic gonadotropin injection. Vasectomized males. To induce pseudopregnancy, females are mated with males that have been vasectomized or that are sterile. That is, the male can mate with the female normally and generate a copulatory plug, but will not deposit sperm. Often these males are known as duds, since the fertile males are called studs. Vasectomized males may be prepared in the facility. Vasectomized and sterile males can also be obtained from a vendor. Before using vasectomized males, it is prudent to test mate them to ensure that the vasectomy was successful. This can be done by mating them to normal females and checking for pregnancy 14 to 21 days later. Many transgenic facilities maintain their own colony of vasectomized or sterile males needed to make females pseudopregnant. The sexual performance of the males should be monitored by plug checking, and the males should be replaced when their performance is deemed unacceptable. That is typically with no plug observed in 3 or 4 times out of 5 matings. Vasectomized or sterile males may be kept up to 8 to 12 months of age before being replaced. A good practice is keep a mix of variously aged males. Thus, vasectomy surgeries or shipments of vasectomized or sterile males should be scheduled on a regular basis. For creating genetically engineered mice. Pronuclear injection. Pronuclear injection introduces the genetic components directly into an embryo as shown in Figure 10.1 in the manual. During the first 12 hours or so after fertilization, the mouse egg contains 2 nuclei, one nucleus contributed by the sperm and the other belonging to the ovum. These 2 nuclei are referred to as pronuclei because they are precursors of the zygote's nucleus. The 2 pronuclei fuse, forming the nucleus. Many copies of the transgene DNA are injected into one or both pronuclei by means of an extremely fine glass pipette. The transgene is incorporated into the chromosomes through the action of enzymes normally present in the cell nucleus. The pronuclei may integrate one DNA fragment, multiple copies of the fragment, or none. These fragments are inserted at random locations on the chromosomes. Following pronuclear injection, the fertilized egg is implanted into a recipient female for gestation, where it will grow as mediated transgenesis. A second method to introduce DNA into an embryo is called transposon-mediated transgenesis. Because multiple copies of a DNA sequence can sometimes be detrimental, scientists use a bacterial system that can introduce the transgene in a single copy. In this instance, the transgene is flanked by special DNA sequences known as transposons, which are obtained from bacteria. Pronuclear injection introduces the transposon-flanked DNA sequence along with an enzyme called a transposase that inserts a single copy of the DNA into the genome. Because of the nature of transposons, they can only hold a small DNA sequence, typically only several thousand base pairs. So this type of transgenesis can only be used to insert small-mediated transgenesis. Another technology is to insert the DNA into embryonic stem cells. These cells are then injected into a mouse embryo at the blastocyst stage to generate an animal that incorporates this DNA in many of its cells. Embryonic stem cells are undifferentiated cells that have the ability to develop into any kind of cell in the body. Embryonic stem cells can divide, differentiate, and develop into an entire animal. Embryonic stem cells are harvested from the inner cell mass of blastocysts, a pre-implantation stage of developing early embryos, and are grown in culture to produce large numbers of cells. Following their genetic manipulation, which is described later, these embryonic stem cells are injected into another blastocyst. Certain mouse strains have embryos that are too fragile for pronuclear injection, or that will fail to develop through all stages of gestation after infusion into a recipient mother, such as bulb C and many 129 strains. For these mouse strains, transgenic mice can be produced using embryonic stem cell-mediated transgenesis. In these instances, the DNA is introduced into mouse embryonic stem cells using a procedure called electroporation. In electroporation, a solution of the DNA is mixed with the mouse embryonic stem cells, and then an electrical shock is applied. This opens pores in the cellular and nuclear membranes so the DNA can flow into the nucleus. There, the DNA can insert itself randomly into the genome. Embryonic stem cells are selected for the presence of the DNA using resistance to antibiotics, which will be described later. Cells with the transgene are then injected into mouse blastocysts, which develop into the desired transgenic animal. More on blastocyst injection of embryonic stem cells will be discussed in the section on targeted mutation. Because classical transgenesis is random, the insertion of the transgene could have occurred in another essential gene. Thus, many transgenic lines are maintained as heterozygous lines that are outcrossed to wild-type animals at each generation. The need for multiple independent lines for the same gene and the need for wild-type animals for crossing means that many animals are used for any one project. As well, you ensure that careful record-keeping is essential to ensure that the mice are mated and genotyped properly. The alteration in production of specific proteins can result in a change in the animal's phenotype, potentially altering the animal's appearance, behavior, physiologic functions, and biochemical parameters. These phenotypic changes are studied to enhance the understanding of disease processes. Because inbred strains are very uniform in their phenotypes, it's easier to see phenotypic changes in a transgenic or gene-targeted mouse from an inbred strain, which is why inbred strains are most commonly chosen for genetic engineering and its impact. You'll note that with each of these technologies, the transgene DNA is inserted randomly into the genome. The location of gene insertions can have a significant effect on the expression of the transgene, for example, whether or not a protein will be produced from it. Pups born after injection of a transgene must first be genotyped to make sure that the animals produced have integrated the desired transgene. The mechanical trauma of the pronuclear injection may cause irreparable damage to the embryo, which may die during development. Most will not incorporate the DNA construct in their genome, as typically only about 20% of pups born will contain the transgene. The researcher then genotypes each offspring by taking a small piece of tissue, such as the tip of the tail, an ear punch, or buccal swab, and performs a PCR, or polymerase chain reaction, analysis to determine if the transgene is present. This process of genotyping identifies which animals have the transgene inserted in their genome. To contain the transgene should be treated as the founder of a separate line of mice, since each should have the insertion at a different site in the genome. The initial founder mouse of each line is labeled F0. Founders should not be bred with each other. Instead, they are bred with wild-type mice to establish a mouse line. Thus, after the F0s are identified as having the transgene, they are mated to wild-type animals to identify those that have integrated the transgene into their germ cells and that will pass it on to their offspring. The offspring of each original founder should then have the transgene in the same place in the genome as its parent. As a result, the next generation, which is the F1 generation, can then be tested to see how well the transgene is expressed. Each subsequent generation of genetically altered to wild-type mating is correspondingly called F1, F2, and so forth. Because mammals carry 2 copies of each chromosome, they carry 2 copies of most genes, one from each parent. These gene copies are called alleles. Recall that when these alleles are identical, the animal is homozygous for that gene. When these alleles are different, the animal is heterozygous. A transgene is generally incorporated into only one of the chromosomes in a pair. This means there is no corresponding copy, an allele, on the matching sister chromosome. These animals are said to be hemizygous, as hemi from Greek means 1/2. In hemizygous animals, half of the germ cells contains the transgene and half contains a wild-type chromosome that lacks the transgene. The results of breeding 2 hemizygous animals are shown in Figure 10.2. When 2 hemizygous animals are mated, 25% of the offspring will have 2 copies of the transgene, which makes them homozygous for the transgene. 50% will be hemizygous for the transgene, and 25% will have no copy of the transgene. In practice, however, the yield of homozygous offspring may be lower because the presence of 2 transgenes may cause congenital defects or even be lethal in the animal produced. If the pups are viable, offspring homozygous for the transgene can then be mated to each other to establish the line. Creation of a single transgenic mouse line can take a year or longer and cost thousands of dollars in technical labor and per diem fees. These extremely valuable and irreplaceable animals must be cared for by professional husbandry staff led by knowledgeable supervisors. The LATG's role is critical in providing staff education and direction. Generating gene-targeted mice. Gene-targeted mice, knockouts, and knock-ins. The power in using genetically engineered mice is specifically to remove, change, or replace genes in the mouse genome. This technology is called gene targeting, and most of the recent animal models being developed take advantage of this technology. For example, one of the first gene-targeted models defined a tumor suppressor gene, which then disabled facilitated tumor development in mice. Mouse models have been developed that replace the mouse immune system with a human system, which can be used to examine how it reacts when challenged with human pathogens. Depending on whether the transgene is a disabled or functional gene, the animals are called knockouts or knock-ins. Knockout animals. The fragment of DNA inserted through gene-targeted technology may prevent the gene product from being expressed. Such mutant animals thus produced are called knockouts. Preventing a gene from being expressed provides valuable clues about what that gene normally does. Since humans share many genes with mice, observing the characteristics of knockout mice allows researchers to better understand how the gene may cause or contribute to disease in humans. Knockouts are used as animal models for the study of cancer, diabetes, arthritis, Parkinson's disease, and many other human diseases. Knock-in animals. A knock-in animal contains a targeted section of new DNA that has been designed to change a specific gene, generally by creating a mutation suspected of contributing to a disease. As an example, a knock-in mouse may contain a mutation in its DNA that causes a known cancer-causing gene, which is called an oncogene, to change its pattern of expression. In turn, the oncogene may become more or less aggressive at causing tumors. In some instances, knock-in mutations have changed the pattern of cancer development so that the tumor is better able to metastasize, that is, to disseminate cells to other areas of the body to form new tumors. Generating gene-targeted mice. Gene-targeted mutations have classically made use of embryonic stem cells, which are undifferentiated cells and have the ability to develop into any kind of cell in the body. Embryonic stem cells are harvested from the inner cell mass of blastocysts, which is a pre-implantation stage of developing early embryos. The embryonic stem cells are grown in culture to produce large numbers of cells. The cells are then grown in the presence of antibiotics, and surviving cells are ones that have taken up the DNA with the resistance gene. Embryonic stem cells are used to make gene-targeted mutations because millions of cells can be easily assayed to find the 1 in a million homologous recombination event. The surviving embryonic stem cells are tested to identify the ones that contain the mutated DNA in the correct spot in the genome. Cells testing positive are then injected into embryos at the blastocyst stage to eventually generate the mouse line. During embryonic development, the mutant embryonic stem cells mix with the cells already present in the inner cell mass of the blastocyst to generate a mouse in which some of the cells are derived from the embryonic stem cells and some of the cells are from the recipient blastocyst. Because the resulting animal is a mix of cells from different animals, it is called a chimera. A technique that allows investigators to follow the contribution of embryonic stem cells to each offspring is to use embryonic stem cells that have one coat color gene injected into blastocysts from mice containing a different coat color gene. Figure 10.3 in the manual shows a typical result from injection of embryonic stem cells from an agouti 129 strain into donor blastocysts from the black C57 black 6 strain. If the procedure works properly, pups born to the surrogate mother will be chimeric. That is, they will have tissues, including skin, that are derived from both the injected embryonic stem cells and the donor cells from the blastocyst. A chimera has patches of fur derived from the embryonic stem cells and the donor. As illustrated in Figure 10.3 in the manual, a B6-129 chimera has agouti fur from the 129 embryonic stem cells and black fur produced by cells from the C57 black 6 blastocyst. Pups that did not incorporate the embryonic stem cells will be all black since they developed only from host cells of the C57 black 6 donor blastocyst. In general, the percentage of fur that comes from the 129 embryonic stem cells is a good indicator of the chimera's subsequent ability to provide germline transmission of the embryonic stem cells and thus the targeted mutation. Therefore, the best chimeras will appear to be all or mostly agouti, being derived from embryonic stem cells with very little to no contribution of the coat color from the blastocyst. The sex of the chimera is determined by both the sex chromosomes of the embryonic stem cells and those of the blastocysts into which they are injected. The embryonic stem cell lines used most commonly are male, X and Y. The blastocyst used may be either male or female. A male embryo recipient that is injected with male embryonic stem cells, both of which have XY chromosomes, will develop into a male. However, if the embryonic stem cells from a male are injected into a female embryo, the resulting mouse will have a mixture of cells, some with XY chromosomes from the embryonic stem cells, and some with XX chromosomes from the embryo's own cells. If this female embryo has a high number of cells derived from the embryonic stem cells, XY, in the gonads, its phenotype may be male because of the sex hormones produced by the embryonic stem cells. If there are only a few male embryonic stem cells in the gonads, this animal may be phenotypically a female, or may be a hermaphrodite, but may be sterile. Classical gene targeting is generally a lengthy process, with the tissue culture steps taking several months and the injections and breeding to establish mouse lines taking another 6 months. Due to the tissue culture involved, it is also quite expensive. Thus, investigators have long been looking for new methods to generate precisely mutated animals. The search has led to the development of engineered systems that will cut the DNA in a spot chosen by the investigator. Once a double-stranded cut in the DNA is made, the cell takes over and causes either a deletion or, if supplied a DNA template, an insertion at the site of the cut. Such systems can use pronuclear injection to introduce the components directly into the embryo. Since they remove the requirement for embryonic stem cell culture, they reduce both time and cost. One of the most commonly used systems is the CRISPR-Cas9 system. Germline transmission. As noted earlier, mice produced from injection of embryonic stem cells into the blastocyst will generate mice that have a mixture of cells from each of these donors. These animals are known as chimeras. In chimeric animals, the gene-targeted mutation is present only in those cells derived from the injected embryonic stem cells. It may or may not be present in the germ cells that give rise to the ovum and the sperm. In the gonads, there could be a mixture of germ cells derived from the host and from the embryonic stem cells. However, the targeted mutation must be incorporated in the germ cell of the chimeras in order to be transmitted to the next generation of mice. This is called germline transmission. The percentage of embryonic stem cell line pigmentation, such as the percentage of brown or light color compared with black, is used as an estimate of the percent contribution of embryonic stem cells to the tissues of the body, including the gonads. Thus, if an animal appears to have 90% of its coat color derived from the embryonic stem cells, it has a high likelihood of germline transmission of the gene. That being said, even animals which have a low percentage of embryonic stem cell line pigmentation may have successful germline transmission. Chimeras are mated to animals of the same strain as the blastocyst host to test for germline transmission. Pups which receive the embryonic stem cell chromosomes will have a copy of the mutated gene in every cell in their bodies. When a chimeric male, for example one that has 129 embryonic stem cells injected into a blastocyst that was C57 black 6, is mated with a black female, such as a C57 black 6 mouse, finding agouti pups in the litter proves that the male chimera produced sperm cells from the embryonic stem cells that were injected, as depicted in Figure 10.4 in the manual. Male chimeras are preferred for breeding over female chimeras unless male chimeras were not produced. If male chimeras are determined to be sterile or do not pass the gene through their germline, that is, repeated mating did not produce pups or produced a succession of litters without any progeny receiving the targeted gene transmission, they are culled. It is important to remember that the embryonic stem cells injected were diploid, and typically only one allele is mutated. Thus, mice arising from a cross of a wild type with a chimera could exhibit germline transmission of the embryonic stem cell, but since sperm are haploid, they may transmit the wild-type copy rather than the mutant copy of the gene. Thus, even germline pups need to be genotyped to see if they carry the mutation. Once germline transmission is obtained, then lines can be set up to generate more pups as well as homozygous lines. Heterozygous mutant mice still have one functional copy of the gene being targeted, which is often sufficient to produce a normal phenotype, and so these mice may or may not have an altered phenotype. The most interesting and informative phenotype is generally produced by mating heterozygous gene-targeted mice to produce some mice that are homozygous for the mutation, meaning that they have no normal copies of the targeted gene. These homozygous mice can be severely affected and may even die in utero or shortly after birth. The lab animal technician can play a critical role in breeding such mice by carefully observing the phenotypes of pups and alerting the researchers to any unusual findings. Additionally, these mice require special and attentive care. CRISPR-Cas9. Systems have been developed that allow for specific gene targeting using pronuclear injection. These systems rely on enzymes that can be directed to a specific target in the genome. The most commonly used system is CRISPR-Cas9, an abbreviation for clustered, regularly interspaced, short palindromic repeats with CRISPR-associated protein 9. CRISPR is a DNA identification mechanism that is naturally occurring. It is the antiviral immune system for bacteria. Upon recognition of foreign nucleic acid, its DNA is targeted for cutting by the associated nuclease Cas9. CRISPR-Cas9 has the potential to repair genetic damage, introduce a mutation, or selectively knock out genes of interest. For researchers, this mechanism is valuable because it allows them to cut DNA at a specific site and therefore edit the genome. The nuclease is brought to the specific site by use of guide RNA sequences, and the DNA is cut. The cell then repairs the cut by one of two methods: degrading the DNA at the cut, removing the gene and creating a knockout, or incorporating a new DNA fragment provided by the researcher to create a knock-in. CRISPR-Cas9 acts as a gene editor with the ability to repair genetic damage. However, because mammals carry 2 copies of each chromosome, they carry 2 copies, alleles, of most genes. Occasionally, with CRISPR-Cas9, both alleles are targeted. If only one allele is mutated, the animal is considered a mosaic animal. To ensure germline transmission of the CRISPR-Cas9-induced mutation, founders are back-crossed to wild-type animals, and the genotype and phenotype of their progeny are assessed as normal. Assessing genotype. You'll recall that the genotype of an animal is an analysis of the DNA of that animal. For genetically modified animals, this would entail checking for the DNA modification that the investigator expects via genotyping. The phenotype of the animal is the physiological modifications that occur in response to the genetic modification, often the visible characteristics of the modified gene. You can see from this discussion that it's very important to identify those animals that contain the mutation. An LATG will often assist the investigator by placing an identification mark, such as an ear clip, tag, or tattoo, on each pup and then isolating some of its tissue so that it may be assessed for the mutation. The typical source of tissue used is a short piece of the mouse's tail. Alternatively, the disc of ear tissue taken by an ear clip or cells obtained by a buccal swab may provide sufficient tissue for genotyping. A tail biopsy is usually done around 2 weeks of age. The very tip of the tail is collected, only 1 to 2 millimeters is necessary for PCR. This procedure is considered painful, so appropriate anesthesia and analgesia should be provided. It is customary to determine the sex of the young mice and biopsy them for genotyping just prior to or at weaning. It is possible to biopsy pre-weaned pups, but there is some risk of disturbing the litter, and the dam may cannibalize the pups if done before 10 days of age. As well, it may sometimes be necessary to take a sample from a mature mouse, perhaps to confirm earlier findings. A tail-tipped mouse of any age should be monitored to ensure that the blood coagulates quickly and the tail tip ceases to bleed. If an adult mouse must be genotyped, this is a minor surgery and will require use of anesthesia and pain medication. Assessing phenotype. In order to determine how mutations in a gene affect an animal's strain of interest, the scientist or the veterinarian will examine mutant animals for the phenotype produced by the DNA change due to genetic engineering. Many institutions have a phenotyping core. While some phenotypes may be easily seen, as in the case of developmental abnormalities, a thorough assessment is required to completely document the phenotype. The technologist is an important contributor to this effort, both in helping to identify animals in need of assessment and in conducting the tests used to characterize the phenotype and genotype. Phenotype may be assessed by a variety of tools and techniques, such as the following: clinical examination to evaluate the animal's general physical condition, assessment of reproductive performance and fertility, neurological tests to evaluate motor and sensory functions, behavioral test to evaluate one or more specific types of behavior that indicate a phenotypical effect, physiological tests to assess data from multiple physiological parameters, imaging techniques to gather data using non-invasive technology, necropsy and histopathology studies to evaluate specific tissues of interest, and clinical chemistry to evaluate function of the various organ systems. While many investigators will do their own phenotyping, specific protocols have been published, and there are specialized centers that will perform phenotyping of any mutant mouse strain submitted. Changing the background strain of mutant mice. On occasion, an investigator may have generated a mutant line in one inbred strain, but needs to move the gene into another inbred strain. This may be done either to make it less detrimental to maintaining the strain, or sometimes to move it into a strain background that is more conducive to obtaining the needed phenotype. For example, a mutation may cause embryonic lethality in a C57 Black 6 strain, but when moved to a bulb C strain, the embryos are now viable. Or the investigator may wish to move a mutation in C57 Black 6 to FVB, where it is more cancer-prone. To change the strain background, each generation of genetically modified mice is bred to wild-type mice from the desired strain. This allows the researcher to gradually dilute out the portions of the genome that carried over from the strain from which the embryonic stem cells were produced. This process is known as backcrossing. Each backcross generation is given an N number, for example N1, N2. This is in contrast to the practice of identifying the progeny of brother-sister matings by an F number, F1, F2, F3. Once a mixed strain has been backcrossed for 10 generations, it is considered essentially genetically identical to the inbred strain, except for the desired genetic modification. It is then referred to as a congenic strain. It's always important to be aware of the genetic strain of a mouse, as different strains have defined mutations that will be present simply due to the strain background, in addition to the presence of artificially produced genetic modifications. Cryopreservation. Cryopreservation is a technique that is widely used in the field of genetic engineering. Cryopreservation of embryos and sperm is useful for recovery of the strain when needed. Less commonly, ovaries may be cryopreserved as a source of oocytes. If live mice of a particular strain will not be needed for some time, it may be advantageous to freeze the line and take the mice off the shelf. If mice are bred within a small colony for many generations, genetic drift may occur due to the gradual accumulation of random mutations. Thus, it's important to cryopreserve a strain early after its isolation. Perhaps the most important argument for cryopreserving genetically modified mouse strains is to allow the colony to be repopulated if the live mice have to be euthanized to eliminate some pathogen from the vivarium, if there is another type of catastrophe that destroys the line, such as a fire or flood, or if they stop breeding successfully. If a mouse line is unique and not banked for cryopreservation, it may be extremely difficult and time-consuming or impossible to recreate the exact genetic modification. The cryopreservation of embryos involves collecting fertilized embryos from donor females. Often the females are wild-type because it's more efficient to purchase wild-type females of the optimum age for superovulation. When the females are mated with hemizygous or heterozygous males, only half of the embryos, on average, will have the desired genotype, while matings with homozygous males will result in all of the embryos having one copy of the modified gene. The embryos are incubated in a solution that protects them from the harmful effects of freezing and stored in screw-cap vials or semen straws in liquid nitrogen. At the extremely cold temperature of liquid nitrogen, embryos will remain viable essentially forever. When mice of a particular line or genotype are needed, the embryos are carefully thawed and surgically implanted in a pseudo-pregnant female for gestation. This is often referred to as recovering a mouse line. Embryo cryopreservation is a very successful method for providing viable embryos in many strains. Sperm cryopreservation is a terminal procedure that requires only one or two fertile males. Procedures for sperm cryopreservation have been developed to the point where virtually all strains can be recovered after thawing the sperm and performing an in vitro fertilization. Perhaps the biggest drawback with sperm cryopreservation is that only half of the germ plasm is banked. Thus, upon recovery, the animals resulting will be heterozygous. Generally, this is an acceptable caveat for most genes. However, if a line has two or more modified genes, or genes that must be maintained in the homozygous state, sperm cryopreservation would not be the optimal method of banking the line. Thousands of genetically altered mouse strains have been created in hundreds of laboratories around the world. It's not possible to house many animals in one place and keep them available for distribution and research. Cryopreservation is used to maintain the strains while minimizing genetic drift and contamination, guarding against disasters, and fulfilling the principles of the 3 R's of replace, refine, and reduce. Thus, various groups and laboratories have created repositories to maintain control over the burgeoning mouse lines and animal numbers. Prominent among them are the Mutant Mouse Regional Resource Centers, which accept submissions of valuable genetically modified murine lines and embryonic stem cells to breed, distribute, and cryopreserve. As the techniques of cryopreservation become more widespread and uniform, the trend will be to maintain more strains strictly in the form of frozen embryos or sperm, and repositories will do only sufficient breeding to replenish the frozen stocks. Summary: genetic engineering is an important tool in animal research. By allowing biological systems to be manipulated at the genetic level, researchers can fine-tune their studies to address complex questions directly. By allowing more specific questions to be answered, the numbers of animals used can be reduced, experimental conditions refined, and potentially animals can be replaced. Moreover, genetic engineering of mice is becoming more and more frequent, with the techniques we mentioned previously being commonplace in many animal facilities. As such, an LATG should have a thorough grounding in the additional requirements that genetically engineered animals have and the methodologies used to produce them. Chapter 11. Nautobiotics. Nautobiotics, which means well-known life, is the study of animals whose microflora is completely defined. That is, the scientists know exactly which, if any, bacteria, fungi, protozoa, and so forth live on and in nautobiotic animals. Germ-free, also known as axenic animals, are only one type of nautobiote, those free of all demonstrable microorganisms. Another type of nautobiote is a defined flora animal. Defined flora animals are animals that were originally germ-free but were given a known group of bacteria and/or other organisms to aid their digestive functions. Nautobiotic animals must also be distinguished from specific pathogen-free animals. Specific pathogen-free animals have normal flora but are free of particular organisms, usually pathogenic, but they are not nautobiotes. The first record on raising germ-free animals involved guinea pigs in 1895. The first germ-free rat colony was established in 1946. Prior to the late 1950s, germ-free research was performed in rigid isolators made of stainless steel and glass. These isolators were large, heavy, and expensive. In 1957, the introduction of the Trexler flexible film isolator system marked the beginning of modern nautobiotic research. The first commercially raised nautobiotic rats and mice became available in the early 1960s. The practical applications of flexible film isolators had a profound effect on the quality and variety of animals available for research. A great deal of the credit for the advancements in nautobiotics must go to the scientists and technicians of the Laboratories of Bacteriology, University of Notre Dame, more commonly known as the Lobund Institute, who developed the first practical and functional germ-free animal models. An ILAR-sponsored workshop held at the Lobund Institute in 1960 was one of the most important developments in the history of laboratory animal medicine. Conducted by Dr. Philip Trexler, the workshop taught 11 animal suppliers how to rederive their animal lines into a germ-free state. This allowed the animal suppliers to rid their stocks of a large number of bacterial, viral, and parasitological disease agents and to offer germ-free animals to the biomedical research community for the first time. The demand for axenic and nautobiotic animals increased significantly after the NIH launched the Human Microbiome Project in 2007, which dedicated $140 million to characterize the human microbial genome. The microbiome refers to the thousands of microbial species that inhabit a specific host. These include bacteria, viruses, fungi, protozoa, and parasites. Interestingly, it's been established that these microorganisms outnumber the host in respect to human cells by a factor ranging from 10 to 100. Studies with rats, mice, and ferrets have proven to be indispensable to our understanding of how the microbiome profoundly affects initiation and progression of diseases such as diabetes, autoimmune diseases such as lupus, inflammatory bowel disease, and arthritis. These studies have the potential to revolutionize the way these diseases are treated as well. The majority of nautobiotic work is performed with rodents. However, pigs, dogs, goats, chickens, and rabbits have also been used. As the demand for nautobiotic animals has increased, so has the need for knowledgeable animal technicians trained on nautobiotic technology. Working with germ-free or nautobiotic animals is unlike typical rodent husbandry, and strict adherence to standard operating procedures is crucial to the success of these studies. Equipment for nautobiotic maintenance. Isolators. An isolator system is a device that creates a barrier between animals and the outside environment, either by preventing microorganisms outside the system from entering the isolator, which is exclusion, or by confining potentially harmful microorganisms within the system, which is containment. An isolator can be as small as a filter-covered mouse cage or as large as a room. It can be made of any material that is impervious to the passage of microorganisms. Rigid isolators are constructed of stainless steel or plexiglass. Flexible isolators are usually made of clear polyvinyl or polyurethane plastic film. Semi-rigid isolators may be made of impact-resistant material, such as polypropylene, on all but one side, and have a flexible film such as polyurethane on the front side. Seams of film material are typically welded for a complete and strong seal. Flexible film isolators are considerably less expensive than stainless steel isolators and can be manufactured economically in many sizes and shapes, as shown in Figure 11.1 in the manual. These isolators make nanobiotic studies financially feasible for virtually all research institutions. Isolators must provide a means by which animals, cages, and supplies can be manipulated inside the isolator, usually with built-in neoprene gloves, while maintaining a barrier. Because these gloves cannot be replaced while animals are actively housed, careful inspection of the fingers and shoulder areas for wear, tears, or punctures prior to every use is paramount to maintaining the integrity of the sterile environment. Isolators must also have a port with an inner and outer seal to allow various sterile materials to pass through without risking contamination of the animals. The isolators must undergo 3 to 6 weeks of consecutive negative testing before animals are housed in them. Testing includes multiple aerobic and anaerobic cultures of shelves, cages, food, bedding, water, and mold trap. The mold trap is a compilation of food and water. Breeding isolators may hold 12 or more cages, while experimental isolators average 3 to 5 cages. To ensure continued sterility of the isolator, materials and mice are tested each time the port to the isolator is opened. Fresh fecal samples are taken, as are swabs from the mouth, fur, and anus. Scheduled sentinel screening also validates that the integrity of the animals and isolator are maintained. All isolator systems, regardless of size or type, must provide temperature and humidity control, a means of controlling the light-dark cycle, a source of sterile air, and a means of exhausting used air. Inside the isolator, temperature, humidity, and oxygen carbon dioxide levels must be fully controlled. The isolator may operate with either positive or negative pressure with respect to the room. For biohazardous agents, additional equipment is required to couple the exhaust air to a central building exhaust to avoid room contamination. Microbiological containment allows units containing different microbiological conditions to be placed side by side with no risk of cross-contamination. Isolator systems. Isolator systems must provide a means by which animals, cages, and supplies can be manipulated inside the isolator. Also, materials from the outside must be transferred safely to the inside without contaminating the animals and their immediate surroundings. This section describes the basic components of isolator systems. Supply cylinder. As shown in Figure 11.2 in the manual, at the entry port is a stainless steel supply cylinder in which materials and supplies are sterilized before being introduced into the isolator. These cylinders are available in various sizes for use with different isolators. A cylinder has holes covered with filter material which allow steam or gas to pass into the cylinder and sterilize the contents. After the isolator supplies are placed inside the cylinder, the opened end is covered with a thin sheet of autoclavable mylar plastic and then taped shut with autoclavable mylar tape. The cylinder and its contents are then autoclaved or gas sterilized. Transfer sleeve. A transfer sleeve is a flexible plastic tube of the same diameter as the entry port and the supply cylinder. The transfer sleeve serves as a transition between the entry port and another sterile container or isolator, thereby aiding the entry or exit of items. Such items may be supplies, waste, and animals. All items to be moved in either direction are first bagged or boxed. After removing the entry port's cap, one end of a transfer sleeve is connected onto the supply cylinder by taping or clamping it all around. See Figure 11.3. The transfer sleeve has one or more nipples closed by a plastic plug. These nipples are opened for injecting sterilant into the sleeve interior. Alternatively, prior to the sleeve's placement, its sleeve interior may be swabbed with sterilant applied by a brush or washcloth. A suitable contact time is allowed for the sterilant to destroy all surface microorganisms. Once the transfer sleeve's connection has been sterilized, use the built-in rubber gloves of the isolator to remove the entry port's inner cap. Then, puncture the mylar cover on the open end of the supply cylinder. Still using the isolator gloves, pull the items from the sterile chamber through the transfer sleeve and into the supply cylinder, and finally to the interior of the isolator. The sterile connection of the transfer sleeve maintains sterility of all items and prevents their contamination by the outside environment. A transfer sleeve can be used for moving items from the isolator to an adjacent chamber, applying the previous steps in reverse. Waste materials or weanling animals may be removed from the isolator without a transfer sleeve. Laminar flow cabinets. Laminar flow cabinets, or laminar flow racks, provide a cage or rack environment with a large volume of air that is filtered, sterile, and in a laminar flow. This air treatment removes airborne microorganisms from the immediate cage environment, thus preventing microbial contamination. Under rigidly controlled husbandry procedures, nautobiotic animals can be maintained in this type of system for many months. In recent years, laminar flow units have come into widespread use because animals are more accessible in them than they are in isolators. Some units hold only a few rodent cages, but room-sized units can hold several hundred cages. Sterilization. Sterilization, which is the complete killing of all living organisms, is crucial for the successful maintenance of nautobiotic animals. Sterilization can be accomplished by a number of different methods, the best known being the process of steam autoclaving. Sterilization methods commonly used in the support of nautobiotic systems include steam autoclaving, dry heat air, peracetic acid of 2% weight per volume concentration, ethylene oxide, gamma irradiation, membrane filtration, which is sterilizing liquids by passing them through filters with pore sizes that will not permit the passage of bacteria or larger contaminants, chlorine dioxide, and hydrogen peroxide gas plasma. Nautobiotic methodology. There are two broad areas of nautobiotics in laboratory animal science. One is the study of axenic animals, which are completely free of all demonstrable microorganisms. Basic research with axenic animals involves studies in nutrition, immunology, lifespan and aging, and other important areas of medical research. The presence of microorganisms in such animals renders these animals non-axenic and compromises research findings. The other major area of nautobiotics concerns the application of barrier housing techniques. The process of eliminating pathogenic organisms through hysterectomy rederivation, or embryo transfer combined with barrier rearing, has resulted in the wide availability of high-quality, disease-free mice and rats and, to a limited extent, other laboratory animal species. Applied nautobiotic technology has led to the development of protective environments for animals with immunodeficiencies, which are naturally occurring or induced by irradiation or drug treatment. Without protective environments, these immunodeficient animals would rapidly succumb to normally non-pathogenic microorganisms from the environment. Axenic animal physiology. Axenic animals differ in a number of ways, both anatomically and physiologically, from animals that have a microflora. For example, the intestinal walls of axenic animals are thinner than the intestinal walls of animals with a microflora. Axenic animals are often characterized by poor muscle development. Their tissue enzyme levels tend to be lower than those of non-axenic animals. Their immune systems are less well developed. They have smaller lymph nodes, Peyer's patches, and thymuses than their non-axenic counterparts because these organs have not been exposed to antigenic stimulation. Their white blood cell counts are lower. They absorb dietary fats better, have higher blood cholesterol levels, and higher bile turnover rates. Axenic animals have a greatly enlarged cecum that is approximately 5 times normal size, as shown in Figure 11.4. Their cecal contents and feces are more alkaline and contain more water than non-axenic animals. In fact, one of the most common health concerns in axenic mice is cecal torsion due to the large cecum. Axenic animals have slower peristalsis, the wave-like contractions that move food along the digestive tract. Likewise, sloughing of their intestinal epithelium is slower. Axenic animals have a longer lifespan than their non-axenic counterparts do, and in the case of rats, they have a lower incidence of spontaneous tumors. In addition, the basal metabolic rate and cardiac output are lower in axenic rats, and they have smaller hearts, livers, and lungs than non-axenic rats. Nutrient requirements also differ for axenic animals. They tend to have higher requirements for total food and water intake, vitamin K, B vitamins, and choline than non-axenic animals. They have lower requirements for vitamin A and vitamin E, and the amino acids lysine and cysteine. Because certain essential nutrients are destroyed or broken down by high-temperature autoclaving, most notably thiamine and vitamin A, fortified feeds are used for nautobiotes. It's best to select a closed formula diet as the formulation will not change from each batch received. It's also recommended to do nutritional testing on the diet selected to ensure the autoclave process is not removing too many nutrients and the animals are receiving a nutritionally acceptable diet. Deriving axenic animals. Axenic animals are derived from hysterectomy and fostering to an axenic foster mother or by embryo transfer to an axenic surrogate mother. Commercial rodent producers typically proceed through several steps before introducing new disease-free breeders into barrier production facilities. With embryo transfer, females are superovulated and bred to males. Embryos are collected and washed to reduce further the risks of pathogen transfer. Then they are surgically transferred using aseptic technique into the reproductive tract of pseudo-pregnant axenic females located inside a surgical isolator. To perform a hysterectomy derivation procedure, pregnant donor animals of a strain or stock not yet available in an axenic state are anesthetized at term. The uterus is removed by sterile surgical technique and passed through a dip tank containing a germicide and then transferred to a surgical isolator. In the isolator, the fetuses are removed from the uterus, dried, stimulated, and placed on a warm heating pad. After the pups begin breathing and become pink, they are adopted onto the axenic foster mother, whose pups have been previously removed. To maintain genetic integrity, all of the foster mother's pups should be removed. Failure to remove all of these pups poses a great risk for contamination of genetic stocks, especially if they and the fostered pups are the same color. Newborn pups from the foster mother may be overlooked if they are covered with bedding material. At weaning, these pups, if not phenotypically different, would be assumed to be the re-derived strain. Genetic contamination of this type is difficult to detect, and it may take several generations of breeding and testing before it's discovered. For this reason, it is prudent to select a foster mother from a line that is colored differently from the pups to be fostered. The next step in the derivation process is thorough microbiological monitoring of the fostered offspring to ensure that contamination did not occur during the surgical procedure and that those agents known to pass transplacentally have not been carried over to the newly derived pups. Mycoplasma species and Pasteurella species can pass through the placental barrier and infect newly derived pups. Killam rat virus, also known as lymphocytic choriomeningitis virus, can also pass through the placental barrier. These and other agents must be ruled out by monitoring the derived animal's microbiologic status before they can be transferred from the isolator to another barrier. After the axenic state is assured, most commercial producers give the derived animals a cocktail of normal symbiotic or non-pathogenic intestinal bacteria to normalize the gut physiology and anatomy. During the 1960s, Dr. Russell W. Shadler provided multiple animal suppliers with a variety of normal flora bacteria as he was isolating them from barrier-reared mice. This led to numerous so-called Shadler cocktails being used by different animal suppliers to protect nautobiotes by standardizing their gut flora and enhancing their gastrointestinal function. In 1978, an altered Shadler flora was developed and subsequently adopted by all commercial suppliers of rodents to replace the numerous Shadler cocktails, not only so the associating flora could be standardized, but also to add more of the normal flora of mice and rats to the cocktail and to make it easier to monitor for contamination. Animals that have received a cocktail of known microorganisms are called defined flora animals. Foundation colonies of mice and rats are generally maintained in the defined flora state instead of in the axenic state to optimize their litter size. In axenics, litter sizes are reduced due to the increased size of the cecum. This is especially true in germ-free rabbits and gerbils. Offspring from foundation breeders are then transferred aseptically to barrier expansion and production colonies. During each phase of production, extensive microbiological and genetic monitoring is performed. Summary: the influence the microbiome of research animals has over their health and response to disease has now been demonstrated. Nautobiotic animals are the ideal tool to examine how experimental models of infection and cancer interact with the microbiome. This chapter has shown the techniques and equipment required for technologists to be able to produce and care for these animals. Their special needs require a higher degree of care, attention, and knowledge than for conventionally housed animals, requiring special training and strict adherence to sterile techniques and work practices.